So today we will be talking about the lymphoid organs. Lymphoid organs are of two types, primary lymphoid organs and the secondary lymphoid organs. Lymphoid organs play an important role in production as well as maturation of the immune cells. Now, immune cells we had already discussed in the previous lecture. Today we will be talk in detail about the lymphoid organ. So first we will be talking about primary lymphoid organ in this lecture, and in the second lecture we will discuss about the secondary lymphoid organs. So what are the primary lymphoid organs? The organs which I have marked in red are the primary lymphoid organ. Primary lymphoid organs are the thymus and the bone marrow. So primary lymphoid organ plays a very important role for the production of the immune cells. Production of the immune cells, we have the special term for the production of the cells, that is hematopoiesis and the lymphopoiesis, right? You'll remember we have discussed it into the cells of immune system. So hematopoiesis means blood cells. We had already discussed about the blood cells. Many of the blood cells plays a very important role into the immune system. So lymphopoiesis and all this occur into the bone marrow only. So how we are going to study the these organs? We will discuss about that, means where it present in our body. Then we will be talking about the histology and the function. So first we will talk about the primary lymphoid organs. So primary lymphoid organs, as I had already discussed, that these are the site in which lymphocytes develop the ability to specifically recognize antigen and do distinguish themselves from the non-self. This is the most important property about the lymphocyte. We had already discussed about lymphocytes. So what is the most important property of the lymphocyte? That they can recognize or they can distinguish between the self and non-self. So this maturation of lymphocyte occurs into the primary lymphoid organs. Then the next most important function of primary lymphoid organ, lymphocytes develop ability. First thing we have seen. Then second, they are produced, and the next is they are also getting the maturation. Now, what are these? This lymphoid organ includes the bone marrow and the thymus. Hematopoiesis occur into the bone marrow. These are producing erythrocytes, platelets, monocytes, granulocytes, B cells, and precursor of the T cell. The point to be noted that bone marrow, that means our first organ, bone marrow, it is just producing the precursor of the T cells, NK cells, dendritic cell, and the mast cell. T cells start as means T cells when they form, so they become the undifferentiated white blood cells, which is called as the lymphocytes, into the bone marrow. Means first they are going to be formed. All the cells, all immune cells, their factory and their production is going to occur into the bone marrow only, but site of maturation is differing. So where they are maturing is, when we talk about the T cells, they matures into the thymus. So they travel through the bloodstream and they go to the thymus. They will mature into the T cells that defend against the viruses, bacteria, fungi, and other diseases. And we have studied a detailed role of all these immune cells. So today we will start first with the thymus. So we know the best about thymus, and we know it is most important for the T cell. When we were learning about the T cells, we have discussed that T cells got their name as a T cells because of their site of maturation, that is thymus. So thymus is the site of T cell development and maturation. But the point to be noted here that their production is not done into the thymus. Their production is done into the bone marrow only. There is only one factory, that is the bone marrow. When we talk about human thymus, so in the case of the human, the thymus is located behind the breastbone, or we can say it as a sternum, and it is present above the heart between our two lungs. When we talk about fetus, so in fetus it weighs just 15 to 20 gram, and it increases in size when we, means when as we are getting adulthood, so that time it becomes fully functional thymus is 40 grams. So thereafter it slowly atrophies. Note down the term atrophies. It means it degenerates slowly. Degenerates means it becomes smaller in size. So this is about the general location, general discussion, introduction of the particular organ. As I told you that we will talk about all the organ of the immune system in three parts. First we will see where it is situated in our body. Thereafter we will discuss the histology. Then at the end we will be discussing about the function. So histology. Histology means the microscopic study of that particular tissue. So to study what type of cells, what things are there, we need to take a small section of a particular organ. We make the slide and we do the microscopic observation, that is called as the histology. Histology is one of the branch of zoology only, in that we learned about a detailed microscopic anatomy. Anatomy means we see what type of the cells are there in the particular organ and what are the roles of that particular cells, and thereafter we discuss and we make the overall view about particular organ. So we have seen about thymus that it is the site of maturation, development maturation. Only T cell precursor means a very simple thymocyte are formed into the bone marrow, and this travel to the thymus, and their actual development and actual maturation occurs into the thymus. But thymus is very particular about its job. It take care that all the cells will be trained very well. So in this process, approximately 95 to 99% of the T cells dies into the thymus only. It has self-reactivity or no reactivity to the antigen. Means here only and only maturation and development process will occur. It is not like that the thymus will give site for antigen and antibody reaction. Nothing will happen. All this will happen into the peripheral organs, or you can say the secondary organ. Understood? So in the case of the thymus here, thymus will concentrate totally on the production of the high quality of the T cells. So let us discuss about the histology. So when we see the thymus, it has two oval lobes which are further divided into the lobules which are separated by trabeculae. Trabeculae are septums. Okay, it is actually a type of septum which divides the two particular part of a tissue like structure. You might have observed into the tissues that there are septas. So these septas are called as, or all these lobules are separated by the trabeculae. When we see the thymus, you will find that it has the outer cortex and inner medulla. When we see the cortex, it is a tightly packed with the proliferating immature T lymphocytes, while medulla you will find there are mature cells. You can see this is a actual original histology slide diagram. So this image, you can see the cortex region is quite denser as compared to the medulla, right? So why it happens that in the case of the cortex, newly encounter this thymocytes, which has just newly formed from the bone marrow and has traveled through the bloodstream and enter into the thymus will be there, and they will proliferate, they will increase in their number, and in the case of their maturation, their training, their perfection, they will enter into the medulla. So as I told you that everyone will not get chance to become mature, to enter into the bloodstream. Only and only there will be a proper test whether the cell has been formed perfectly or not. So once the cell has completed its development, that time only the cell will be allowed to enter into the bloodstream and to react with the other cells. Now when we talk about the thymus, you will see that the cells of thymus have four important types. Now it will have, you will find thymocytes. Thymocytes are the T cells precursors. Then dendritic cells, epithelial cells, and the macrophages. So when we talk about the framework, thymocytes we will not call about the framework, because these are the cells, or these are the main players which are going to be mature. Then dendritic cells, epithelial cells, and the macrophages. Now what are their roles we will discuss in short. First, when we see about the thymic epithelial cells, thymic epithelial cells are also called as the nurse cells. Now nurse cells, see this is the nurse cell. You can see it has curved, or it has taken some thymus inside it. We will study the role. They have the long membranous processes which enclose more than 50 thymocytes. This is just a diagram which creates a large multicellular complexes. This corticoepithelial cells and the medullary epithelial cells are responsible for education or selective proliferation of the thymocytes. Okay, so when we talk about the epithelial cells, you have to remember the role of the epithelial cells. Thymus, four type of the cells. Thymocytes, main player means their education, their maturation is going on. Understood? Then epithelial cells, we said another name as the nurse cell. Why we are telling nurse cell? Because they are taking care, they are giving the nourishment. Nurse term has become from the nourishment, so they are giving nourishment, they are taking care. So these nurse cells will take some thymocytes. Here I have shown only 3 4, but actually 50, approximate 50 thymocytes they enclose, and they make the multicellular complex. Now how do they do such things? So they are doing it by the four important hormones. Now, name of the hormone you have to remember. This epithelial cell will produce four hormones: thymulin, alpha thymosin, beta 4 thymosin, and thymopoietin. So, what is the role of this hormone? These hormones bring the transformation of lymphocyte to the T lymphocytes. So, this is a very important thing, that the thymus have the nurse cell, and this nurse cell is educating. Now see, they are educating to all the proliferated cells, but thereafter what happens, that medulla will go. So now from here, you can see that these are making the framework. They are having the structure. Now here medulla cells, they will check whether the thymus cells are properly formed or not formed. We have studied the role of the macrophages and dendritic cell. If the cell is not able to recognize the self and non-self, so that time that particular cells are killed. Okay, and they are going to kill that cells, and this macrophages will engulf. There will be no debris. That's why the cortical region is dense and the medulla region is not that dense. It is having only few thymus. That means thymocytes. Now these are not only thymocytes, these are the T cells. So what will happen now? This medulla region we will discuss. The medulla of the thymus is a continuous between the lobules. It consists of vascular structure, reticular epithelial cells, and you can see the lymphocytes are quite scattered here. The reticular cells are relatively more in number than the lymphocytes. Yes, we can see here reticular cells are more, but lymphocytes are very few. In the medulla are interdigitating dendritic cells. These cells are associated with the epithelial network. They form the complete network for the proper checking, and these are rich in MHC class 2 molecules. Why they have this? Because on the MHC class 2 molecules we have discussed they are presenting the antigen. So here they are going to take the test whether they recognize the antigen or not. In addition, there are small masses of the epithelial cells around a central core of the degenerating epithelial cells, which is known as the Hazel's corpuscles. So this is about the cells of thymus. Once again, we will just discuss about the histology. We have discussed that the thymus is a bilobe gland, and these lobes also divided into the lobules, and these lobules are separated by the trabeculae. Two important part. First, outermost is the cortex, and then thereafter medulla. We have seen that the cortex is denser. Why? Because it is having the proliferating T cells. Then we have discussed about medulla T cells are there. Then medulla of thymus is continuous between the lobules. We have discussed it consists of vascular structure, reticular epithelial cells, and the scattered lymphocytes, as well as the small masses of epithelial cells and the Hazel's corpuscles. Then we have discussed about the framework. Framework is made by the epithelial cells, macrophages, and the dendritic cells. We have seen that four type of cells are there. These are the thymocytes and remaining framework cells. So thymocytes are the T cell precursor which are formed or which are produced into the bone marrow and travel to the thymus via blood circulation. And these immature T cells or the thymocytes are trained or guided by the nurse cells, which is also called as the epithelial cells. So what is the role of the nurse cells? These nurse cells are actually long membranous process which includes more than 50 thymocytes and create the large multicellular complexes. These nurse cells have the corticoepithelial and medullary epithelial cells. The most important role of the nurse cell we have seen is the educating of the thymocytes. Means they are making the precursor of T cells into the mature T cells. Now, how do they bring this task? They are bringing this task by secreting four type of important hormones. It is important to remember the name of hormone. These are the thymulin, alpha thymosin, beta 4 thymosin, and thymopoietin. So these hormones brings the changes or the transformation of the transformation of the lymphocytes, or you can say the T precursor cells to the mature T lymphocyte. But here only maturation does not occur. There is a proper testing. The proper testing means they check whether the thymus is properly formed or not formed. So for that particular reason, after this treatment, after the education, the thymus is sent into the medulla. But in the medulla, this framework is made. You can see the reticular cells are, see this diagram. Reticular cells are more, means this all are the testing agencies here. You can say they are going to check this formed thymus or T cells. We have studied about the role of the T cells. Their role is very crucial into the immune system. So for that particular reason, their formation and their perfection is also very important. So for that particular reason, that perfection are being checked. You can see here the interdigitating dendritic cells are there. Interdigitating means they have spread their digits like structure, so that not a single thymocytes can cross the barrier until and unless they will not test it whether it is formed properly or not formed. But if thymocytes means, or now you can say the T cell, because they have got the education from the nurse cell. So if the T cell is failed to recognize between self and non-self, they are going to kill it, and macrophages are going to engulf that particular thing. So for that particular reason, we have seen that out of 95%, only 1 to 5% are becoming mature, and one who have passed the test will allow to leave the medulla and enter into the bloodstream or go to the secondary lymphoid organ. So this is about the histology. Now we will just discuss about the functions of the thymus. We know that thymus is very important in T lymphocytic proliferation as well as cell mediated immunity, because the central player or central cells are T cells. So if T cells are there, so that time only we can see the cell mediated immunity. T lymphocyte proliferation also occurs here. That means only few numbers will be formed or produced by the B cell, into the bone marrow, and when they will come into the thymus, they will increase in the number. So their proliferation occurs into the cortex region. See, if the thymectomy is done, so you will find in that particular organism cell-mediated immune response does not occur. Ectomy means removal of a particular gland or a particular organ. Ectomy, okay, or amputation means cutting half. Ectomy, complete removal of a gland. Okay. So if thymectomy is done, means thymus is removed by operation from a particular organism, you will find in that particular organism cell mediated immune response does not occur. So it clearly shows the role of the thymus, that thymus is very important for the T lymphocytic proliferation as well as the cell mediated immunity. So as thymocytes develops, an enormous diversity of T cells receptor is generated by a random process that produce some T cells with the receptor capable of recognizing antigen MHC complexes. So as I told you that the training is done into the medulla region, they check whether this thymus been formed are perfect or not. As I told you, thymectomy results in impairment of cell-mediated immunity, which leads to some form of fetal infections. Thymectomy is necessary in some cases. As I told you, cell-mediated immune system, they are very much professional in recognizing or means differentiating from self from the non-self. So if in case a person requires the graft transplantation, suppose a person's kidney is not functioning and the person wants to take kidney from the particular donor, so that particular kidney will not be accepted. It is called as the graft rejection. So for the purpose of the graft retention, because particularly at that moment that person's kidney is more important, okay, the person's kidney is not functioning, and for that reason that person has planned for the transplantation. So to maintain that graft, so thymectomy is done. So they remove the thymus, so cell-mediated immune response will not occur in the body, and that graft will retain in the body. So this is all about the thymus. Now we will be discussing about the second organ, that is the bone marrow. As we had already discussed that bone marrow is the site of generation of all circulating blood cells in the adult, including the immature lymphocytes, as well as it is the site of B cell maturation. Point to be noted, B cells will be formed as well as the B cells will become the mature there. Here, how the blood cells form. Blood cells form by the process of the hematopoiesis. We had already discussed about it in the previous lecture. So one cell will come ahead, it will form its precursor cells and it will give the variants. Okay. So this is the process. First stem cells will come and it will give rise to undifferentiated lymphocytes. Then second step, it will form undifferentiated lymphocytes to process into the thymus gland to become the T cells. Some undifferentiated lymphocytes are processed within the bone marrow. It will become the B cells. Both T cells and B cells are transported to the blood for for doing the immune responses. So it will go to the lymphatic organs like lymph node, lymphatic ducts, and spleen. We all are going to discuss in detail about it. So when we talk about the lymphoid progenitor cells, so it develops into the B lymphocytes into the bone marrow only. Some lymphoid progenitors migrate into the thymus. We had already discussed that they develop into the T lymphocytes. When the secondary immune response occurs, so that time large number of plasma cells are produced within the bone marrow. This secretes large amount of antibodies. So bone marrow is nothing but the antibody synthesis. It is also giving. We have discussed about thymus, that thymus is only and only concentrated on its work. What is its task? To mature the T cells. But unlike of this thymus, the bone marrow is not only producing the various type of cells, it is maturing the B cell there only. At time of maturation, if any secondary immune response occurs, so that particular time the plasma cells are also produced there only. See, plasma cells we had already discussed when we were talking about the cells of immune system, that the B cells takes its role when the antigen is having its re-exposure. So that particular time, that B cell takes that particular immunoglobulin on its surface and start proliferating, and it becomes the plasma cells. That means these particular B cells are specific for the formation of one type of antibody. So we can say that the bone marrow is not only the factory of the various cells, but also it is the source of antibody synthesis also. Bone marrow is the site of generation of all circulating blood cells in the adult, including lymphocytes, as well as it is the B cell maturation. Now we will be talking about the histology of the bone marrow. Bone marrow is a soft tissue within the cavity of the bones. Now see, bones are very stiff structure, right? So inside that you will find the soft tissues are there. The internal surface of the bone has several ridges with the shelves. You will find spicules also. So this all form the sponge-like reticular framework of bony tissue. This will called as the trabeculae. As I told you that the any thing which is separating the tissue, continuous tissue, is called as the trabeculae. So the spaces between hollow reticulum are filled with fat cells, collagen fiber, fibroblasts, osteoblasts, dendritic cells, and reticular cells. Osteoblasts, you all may remember, Osteoblasts are the specialized macrophages of the bone marrow. We have discussed when we were talking about the cells of immune system that macrophages can be specialized for the special organ, right? So all these cells associated with the blood cells precursor of the various lineages and the maturity. Bone marrow is richly supplied with the venous and the arterial vessels. Proliferation as well as differentiation of the lymphocytes and other blood cells is stimulated by the various kind of the cytokines. We know that for they required some type of signal. So these signals are nothing but the cytokines. These cytokines are produced by the macrophages, stromal cells present into the bone marrow, and this different cytokine promotes the proliferation and maturation of the different lineage of the cells. When we talk about the mammals, in mammals, few days after secondary response activated the memory B cells which are found to migrate to the bone marrow. Means when we were talking about the B cells, I told you that they make their one personal copy. If any type of cells will come, means any if the antigen is re-exposed, so that time they don't want to do the complete task again. So for that reason they form the memory cells. So where this memory cells resides, this memory cells resides into the bone marrow only. So the bone marrow is also found to be the major site of antibody synthesis. Peripheral lymphoid organ respond rapidly to antigen but only for relative short time, whereas bone marrow starts slowly and gives a long lasting effect. This is one of the most important difference between the peripheral system and the primary system. So what happens, peripheral system have the trained cell. Now we are going to discuss about the peripheral lymphoid organ into the next lecture. So peripheral and secondary are not different. The peripheral lymphoid organ means the secondary lymphoid organ. So you will find that they are very fast acting. Any antigen has come will be cleaned very fast. Means this army and these soldiers are present there. But these bone marrow and thymus, they are the training centers. They are preparing the army. Okay, so for that particular reason, they are slow acting because training takes time, right? So for that particular reason, they are slow acting. And the secondary peripheral lymphoid organ have the trained complete army. So for that reason, you will find that they are fast acting. But this army is changing, keep on changing. So they don't have the memory, right? But bone marrow, they are although slowly acting, but they have the long lasting memory. So this is all about primary lymphoid organ. Next lecture we will discuss about the secondary lymphoid organ. So at this note we'll stop now.